Hello and welcome back for a webinar on general neurology. My dear students, we have started the study of general neurology since 2 days back, and we have completed the introductory part of general neurology, means anatomical aspect of nervous system. We know what is nervous system, what is central nervous system, what is peripheral nervous system, what is autonomic nervous system, and we know everything about the types of cells present in the nervous system, neurons and the neuroglia. So this study we have completed, and today we will proceed further with the study of general neurology, and I'll take you to the second episode today, and this is more basic. So even though it is little what you call tough to understand, it is required in order to understand further the neuroanatomy as far as the human anatomy is concerned, as well as it is required for understanding the nervous system, which is a major system in your physiology. So that is where we will go step by step. So I welcome you all back again for second episode of general neurology today, and we have to complete it within a span of 30 minutes. So let us have patience and kindly avoid disturbances to me so that we can complete this session within time. First, we'll talk about blood-brain barrier. So barrier means like a barricade. What should go to the brain and what should not go to the brain. So that is, for that purpose, you require a barrier. Now there are lots of barricades on the roads also due to corona lockdown measures. Similar kind of barricades are there in the brain to protect the brain. Like certain dyes, when injected intravenously, they fail to sustain the parenchyma of brain and spinal cord, although they pass easily into the non-nervous tissues. You understand this? However, the same dyes, when injected into the ventricles, they enter the brain substance easily. So what should enter the brain substance and what should not enter the brain substance, that is decided by the blood-brain barrier. So that is what we are talking now. The brain is the highest controlling and coordinating organ of your body, and it requires a lot of protection. So from outside there is a good protection. There is a scalp, tough skin of layered skin, and plus there is a tough bony cage which is called as a skull. It's very tough, and then inside again there is a blood-brain barrier. This indicates that a barrier exists at the capillary level between the blood and nerve cell. So where exactly this blood-brain barrier is located? Is located between the blood capillaries and the nerve cell. So there is a layer there. So that layer is called as a blood-brain barrier. Now what this blood-brain barrier consists of? The possible structures constituting the blood-brain barrier are capillary endothelium without fenestrations, then basement membrane of the endothelium. Okay, you have to write this because short note may be asked, can be asked, and the end feet of astrocytes covering the capillary walls. So these are the things which are present in the blood-brain barrier. The the barrier permits a selective passage of blood contents to the nervous tissue, and thus the toxic and harmful substances are ordinarily prevented from reaching the brain. Now this is the reason why we have a blood-brain barrier. I hope you got it, what exactly I mean to say, that brain requires protection, and certain toxic and harmful substances which may cause harm to the brain are prevented from reaching the brain. So for that purpose, you require a blood-brain barrier. Well, here you can see one image. Here you can see there are different, pictures shown in one slide. Blood-brain barrier system shown here. You can see astrocytes, artery. I told you about the capillary, and you can see the neuron here, and see how it is protected. Okay, here you can see blood CSF barrier, because CSF is present in the cranial cavity because it is important fluid which replaces lymph inside the cranial cavity. So blood CSF barrier is shown in this picture. You can see ependymal cell, and you can see the basolateral surface. So it's a brain surface here, and here you can see the ventricle choroid plexus and ependymal cell. Arachnoid barrier, blood retinal barrier towards the eye, because retina is part and parcel of nervous tissue. Blood spinal barrier. So wherever there is a nervous tissue, there is a barrier, and this barrier will protect the brain and different parts of the brain. So this is in short about blood-brain barrier. Now we will go to another subtopic of today's webinar, and that is Replex Arc. Now Replex Arc is known to you because you have already studied this. This is more basic of nervous system. This is already taught in the school days. A Replex Arc is the basic functional unit of the nervous system. Remember, basic functional unit, Replex Arc, and which can perform an integrated neural activity. What it is made up of, that we'll understand now. One, a receptor. That means wherever there are receptors in your body, for example, in the skin, there are receptors of pain, touch, temperature, hot, cold, okay, pressure. So there are different receptors in the skin. So those receptors are required. Then a sensory or afferent neuron, which will carry this sensation to the brain. So it is afferent neuron. Then a motor or efferent neuron coming from the brain for for the action. And the last is the effector cell, which is present in case of say muscle, there are myocytes, so there are effector cells. So that will take final action. So here it is called as a basic functional unit of the nervous system which perform an integrated neural activity. So these four things are required to make a Replex Arc. In the picture you will understand it very clear. I'll go to that point later on. The complex forms of Replex Arc are polysynaptic. And now you know what is synapse. Okay, it is a contiguity rather than connectivity between two neurons or parts of neurons. So the complex forms of Replex Arc are polysynaptic due to addition of one or more internuncial neurons or interneurons in between the afferent and efferent neurons. Afferent and efferent neurons, ascending and descending neurons. An involuntary motor response of the body is called as a reflex action. The stretch reflexes like tendon jerks are the examples of monosynaptic reflexes, whereas the withdrawal reflexes response to a painful stimulus is called as a polysynaptic reflexes. Now, the painful stimulus, okay, and that picture, that image is quite common in the nervous system. I'll show that picture so you understand this. Now, this is your reflex, because here there is a pain, and pain is carried by the sensory receptors, okay, and finally from brain there is a command for a motor neuron for effector organ muscle to take the hand back. So if the hand is burning, you get a pain, so immediately you take a back. So this is the starting of Replex Arc sensation, and motor action is the end of the Replex Arc. This is the provision everywhere in the body as far as response to painful stimulus is concerned. And the stretch reflexes, the body stretches. For that purpose, you require a monosynaptic reflex. These are the basic types of reflex I told you right now. And this provision of motor neuron and sensory neuron, right, starts from the target organ or target cells up to into the spinal cord, and through spinal cord to the brain. So here it is a image of a spinal cord, how the sensory root, motor root is arranged in a spinal nerve. So they make a spinal nerve. So we have studied how the nerves are formed in our earlier study. Now we will go to peripheral nerves. Once again, you will come to understand what are actually peripheral nerves. I hope that you are already started studying upper limb. So now in upper limb, you must have studied about big nerves, at least axillary nerve, radial nerve, and median nerve, ulnar nerve. These are the big nerves. Just keep those nerves in your mind while studying this subtopic of peripheral nerves. Nerves are solid white cords. Now everybody has seen nerve in the anatomy lab, so you must be knowing all about it. And I don't know whether Corona lockdown, has it become possible for you to go to the anatomy lab and see dissection was just started. I heard that. But if not, if someday you will have to go to the anatomy lab and see the nerve by your naked eyes. The nerves are really solid white cords composed of bundles of nerve fibers, and these bundles are called as fasciculi. One bundle is called as fasciculus. So each nerve fiber is an axon with its covering. So lots of nerve fibers make a fasciculi, and lots of fasciculi make one bundle, and that bundle is called as a white bundle, becomes a white cord, and that white cord is a nerve. The nerve fibers are supported and bound together by connective tissue sheaths at different levels of organization of the nerve. The whole nerve trunk is ensheathed by epineurium, each fasciculi by perineurium, and each nerve fiber by a delicate endoneurium. So now the neurium, these are the sheaths around the nerve. The toughness of a nerve is due to its fibrous sheaths. Otherwise, the nerve tissue itself is very delicate and friable. The toughness of a nerve is due to its fibrous sheaths. Otherwise, the nerve tissue itself is very delicate and friable. So the nerve becomes tough because of this sheath. Actual nervous tissue is delicate, friable. Okay. Now we'll go to spinal nerves. The earlier topic also I told you that there are 31 pairs of spinal nerves. Though there are 33 single vertebrae, there are 31 pairs. Okay, they correspond to the vertebrae, but there are some exceptions. Like cervical nerves are 8, whereas cervical vertebrae are 7. Now 12 thoracic nerves correspond to the 12 thoracic vertebrae. 5 lumbar nerves correspond to the 5 lumbar vertebrae, and 5 sacral nerves, they correspond to 5 sacral vertebrae, which is a fusion of 5 in sacrum. But there are 4 coccygeal vertebrae fusion, but only 1 coccygeal nerve. That is why we get 31 pairs of spinal nerves. Area of skin supplied by a single segment of spinal cord is called as a dermatome. So any single nerve going and supplying an area of the skin, that area is called as a dermatome of that particular nerve. Now here you can see all those nerves, 8 pairs of cervical nerves, 12 pairs of thoracic nerves, 5 pairs of lumbar nerves, 5 pairs of sacral nerves, and 1 pair of coccygeal nerve. And you can see the spinal cord here. When the spinal nerves come out, at 2 places they form big plexuses, which are called as nerve plexuses. So spinal nerves to arm, they form a brachial plexus, and spinal nerves to the lower limb, their thigh, they form a lumbosacral plexus. Here again you can see those vertebrae are counted on one side and nerves are counted on the other side. You can see 8 cervical, 12 thoracic, 5 lumbar, 5 sacral, 1 coccygeal, different colors. And I hope now this is understood by you all, because this is more basic. Each spinal nerve is connected with the spinal cord by 2 roots, a ventral root which is motor and a dorsal root which is sensory. The dorsal root is characterized by the presence of a spinal ganglion at its distal end. In the majority of nerves, the ganglion lies in the intervertebral foramina. Intervertebral foramina, we have seen that in a picture of vertebral column. In between, 2 vertebrae, there is a intervertebral disc. Similarly, in between 2 vertebrae, there are foramina for outlet inlets of these spinal nerves. Okay, 2 roots: a ventral root which is motor and a dorsal root which is sensory. Ventral means towards abdomen, dorsal means towards your back. So there are 2 roots, sensory and motor. The ventral and dorsal nerve roots unite together within the intervertebral foramen itself to form a spinal nerve, and then they come out. The nerve emerges through the intervertebral foramen, gives off recurrent meningeal branches, and then divides immediately dorsal and ventral. After forming a nerve, again they divide into 2 ramus. Ramus means branches. They form in a root and they divide again in a branches. So dorsal branch and ventral branch. Dorsal ramus and ventral ramus. The dorsal ramus passes backwards. So back of the backward sides supplied by the dorsal ramus supplies the intrinsic muscles of the back and the skin covering them. The ventral ramus is connected with the sympathetic ganglion and is distributed to the limb or the anterolateral body wall. In case of a typical spinal nerve, the ventral ramus does not mix with the neighboring rami and gives off several muscular branches, a lateral cutaneous branch and an anterior cutaneous branch. Cutaneous means skin, the branch which is going to the skin. Muscular branches means branches which are going to the muscles. However, the ventral rami of the other spinal nerves are plated to form the nerve plexuses for the limbs, like the brachial plexus and lumbar plexus. This is very important because it's asked in exam, brachial plexus and lumbar plexus. The ventral rami of other spinal nerves are plated. We are talking about remaining what we saw in earlier in thoracic, spinal nerves. So thoracic spinal have nerves meant for the sympathetic ganglia chain. Okay, there is a chain, and these are the remaining are forming the plexuses, brachial plexus for upper limb and lumbar plexus for our lower limb or lumbosacral plexus. Here you can see the spinal cord shown one segment. In one segment you can see inside the gray matter of the spinal cord, you can white matter, and from gray matter you can see two nerve roots, getting attached. Posterior rootlets, anterior rootlets. Rootlets get converted into root. So anterior nerve root, posterior nerve root. Anterior means ventral, posterior means dorsal. They unite to form a nerve, and this is actual spinal nerve with a spinal ganglion. And this nerve gets divided into two branches. Posterior ramus means dorsal branch, and anterior ramus means ventral branch. This much of study at least you should know very well on the tip of the tongue, because there is no excuse for this. Okay. Now we'll go to the nerve plexuses, because already we have spoken about two important nerve plexuses, and those are brachial plexus and the lumbar plexus. All nerve plexuses are formed only by the ventral rami. Now the anterior branches take part in the formation of plexus. Posterior branches do not take part in the formation of these nerve plexuses, and never by the dorsal rami. So, okay, there are some spelling mistake, kindly understand. They supply the limbs. So brachial plexus for the upper limb, and the lumbosacral or lumbar plexus is for the lower limb. Against these plexuses, the spinal cord is enlarged to form cervical enlargement for the brachial plexus and the lumbar enlargement for the lumbosacral plexus. At this point, the spinal cord is enlarged. Okay. Each nerve root of the plexus divides into ventral and a dorsal division. Now we saw dorsoventral roots, then we saw dorsoventral ramus. Now here we are seeing dorsoventral divisions. Okay, because now for the purpose of formation of plexus, divisions are required. The ventral division supplies the flexor compartment, and the dorsal division supplies the extensor compartment. Here flexor compartment, here extensor compartment of limbs. Okay, both the limbs. The flexor compartment has a richer nerve supply than the extensor compartment of the limb. Understand this difference between two compartments of limbs. The flexor skin is more sensitive. Now this skin is more sensitive than the extensor skin. The extensor is rough. Okay, the flexor muscles, like they are bulkier because they are anti-gravity and are more efficient and under a more precise control than coarser extensor muscles. So these muscles have this difference due to the nerve supply. Here you can see a brachial plexus coming out from nerves from cervical region, so C5 to T1. This is the brachial plexus eventually giving branches and going to the upper limb. Here you can understand the brachial plexus very well. You can see the roots C5, C6, C7, C8. So 4 of cervical and 1 of thoracic, first thoracic. So T1. So C5, C6, C7, C8 and T1. So 5 spinal nerves are taking in formation of your brachial plexus, and these roots, they get divided into, they form trunks, because this anterior divisions, ventral rami of these nerves, they eventually, when they divide into what you call anterior and posterior divisions, they unite. This is how they unite to form into trunks. The trunks get divided, eventually cords are formed, and cords gives branches which are called as actual nerves. Now this is a what you call regional anatomy study, and that regional anatomy study we will study during the time of upper limb study. So this is just outline of brachial plexus which I am showing you. Now we'll go to other topic, that is blood and nerve supply of peripheral nerves. Peripheral nerves are supplied by vessels called as vasa nervorum. Vasa means vessel, and nervorum means vessels which supply the nerves, which form longitudinal anastomosis on the surface of the nerves. The nerves distributed to the seats of the nerve trunks are called nervi nervorum. So nervi nervorum means nerve which supplies the nerve, and vasa vasorum means vessel which supplies the vessel, and vasa nervorum means vessel which supplies the nerves. So try to understand these Latin words: nervi nervorum, vasa vasorum, vasa nervorum. Okay. Now each nerve fiber is an axon with its covering. Larger axons are covered by a myelin sheath and are termed myelinated or medullated fibers. The fatty nature of myelin is responsible for the glistening whiteness of the peripheral nerve trunks. If you see the peripheral nerve trunks, the cords, they are having a glistening whiteness, and that is due to myelinated or medullated fibers and the white matter of the CNS. The thinner axons of less than 1 micron diameter do not have the myelin sheath and are therefore termed non-myelinated, non-medullated, or gray matter. So gray matter, I showed you in the diagram of that spinal cord, because they are having what you call non-myelinated gray fibers. However, all the fibers, whether myelinated or non-myelinated, have a neurolemal sheath which is uniformly absent in the tracts. In peripheral nerves, both the myelin and neurolemal sheaths are derived from Schwann cells. New types of cells, Schwann cells we have studied in neuroglia. Now we'll talk about myelinated fibers. Myelinated fibers form bulk of the somatic nerves. Somatic nerves means nerve which goes to your body are called as somatic nerves. The bulk of the somatic nerve is formed by the myelinated fiber. Structurally, they are made up of the following parts from within outward. We can see one is axis cylinder forms the central core of the fiber. It consists of axoplasm covered by axolemma. We are trying to understand the myelinated fibers constitution. Axis cylinder forms the central core of the fiber. Then myelin sheath derived from small cells surround the axis cylinder. It is made up of alternate concentric layers of lipids and proteins formed by spiralization of the mesoxon, and the lipids include cholesterol, glycolipids, and phospholipids. And a myelin sheath is interrupted at regular intervals called the nodes of Ranvier, where the adjacent Schwann cells meet. Collateral branches of the axon arise at the nodes of Ranvier. Thicker axons possess a thicker coat of myelin sheath and longer internodes. Each internode is myelinated by a Schwann cell. Oblique clefts in the myelin are called incisures of Smith. Lanterman. So many anatomists have discovered so many small things, and those things are named after those anatomists. Okay. Myelin sheath acts as an insulator for the nerve fiber. And next, third is neurolemal sheath. Neurolemal sheath, sheath of Schwann, surrounds the myelin sheath. It represents the plasma membrane of the small Schwann cell. It represents the plasma membrane of small Schwann cell. Beneath the membrane, there lies a thin layer of cytoplasm with the nucleus of the Schwann cell. The sheath of two cells interdigitate at the nodes of Ranvier. Neurolemal sheath is necessary for regeneration of a damaged nerve. Tracts do not regenerate because of its absence. Now, fourth structure is endoneurium. Inside, endoneurium is a delicate connective tissue sheath which surrounds the neurolemal sheath. Here you can see non-myelinated axon and myelinated axon. So non-myelinated axon, you can see there is no myelin as such. Here you can see the myelin on the axon. Okay. Well, what we have next now? Next is non-myelinated fibers. Okay, opposite of that. Non-myelinated fibers comprise the smaller axons of the central nervous system. In addition to peripheral postganglionic autonomic fibers, several types of fine sensory fibers and olfactory nerve, etc. Structurally, a non-myelinated fiber consists of a group of small axons due currently size 0.12 to 2 microns diameter that have invaginated separately by a single Schwann cell without any spiraling of the mesoxon. The endoneurium, instead of encysting individual axons, surrounds all the neurolemal sheath, by virtue of which non-myelin fibers like the myelinated fibers, can regenerate after damage. So difference between non-myelinated fiber and myelinated fiber is the regeneration. Myelinated fiber will have regeneration capacity after damage, where non-myelinated fiber will not have. Now, classification of peripheral nerve fibers according to the function, the nerve fiber may be somatic efferent because this for supply the striatal muscle of somatic origin, general visceral efferent to supply smooth muscles and gland, and spatial visceral efferent to supply striatal muscles of the branchial origin. Branchial means what you call pharyngeal arches, this region. Brachial is different and branchial is different. Please don't try to make a mistake. Because of N, we get two meanings. Now, general somatic afferent is for to receive impulses from the skin and proprioceptive impulses from the muscles, tendons, and joints. And general visceral afferent to receive impulses from the viscera, just like pain. And spatial visceral afferent is to carry the sensation of taste. And spatial somatic afferent is to carry the sensation of smell, vision, hearing, and equilibrium. There are 4 spatial senses, and the fifth one is a general sensation complex, which includes pain, touch, temperature, pressure, proprioception. Now, all these spatial senses and general senses you have to study nicely in the study of nervous system and physiology more than in anatomy. And I think we have completed this topic on what we call different types of nerve fibers. And according to the size, we'll try to understand size and speed of conduction also. The nerve fibers are divided into 3 categories, namely A category, B category, and C category. Well, in A category group of fibers, they are thickest and faster, for the sake of conduction, thickest and fastest group A fibers, and are myelinated 1.5 to 21 microns in diameter with a conduction speed of 4 to 120 meters per second. So this is a conduction system in the nervous system. So there is a speed for that. Example scale, skeletal-motor fibers, alpha A, or physiomotor fibers, beta A and gamma A. This is in depth as far as the group and the different fibers are concerned. Group B fibers have medium size and medium speed. They are myelinated plus 1.5 to 3 microns in diameter. Okay. Example is preganglionic autonomic efferents. And the last group is group C fibers. They are thinnest and slowest, and they are non-myelinated, 0.1 to 2 microns in diameter with a conduction speed of 0.5 to 4 meters per second. So you see the difference of speed also. And examples are postganglionic autonomic efferents and afferent fibers to skin, muscles, and viscera. The same efferents with a conduction speed and myelination similar to B fibers are called as delta A fibers. Thus, the fiber diameter is directly proportional to conduction velocity. Now, the last part is degeneration and regeneration of neurons. I told you in the beginning that nervous system is very poor in regeneration. Almost, except those supporting cells, there is no regeneration. And plus they go into degeneration. So damage to nerve is almost a permanent loss. If a nerve axon is injured or cut, a series of degenerative and then regenerative changes follow. The degenerative changes in the neuron occurs both proximal, that is retrograde degeneration, and distal, antegrade degeneration to the site of injury. The cell bodies undergo chromatolysis within 48 hours of injury. The cell becomes swollen, rounded, and nucleus is pushed to the periphery, and the nuclear granules disintegrate and disappear. The proximal part of the axon survives if the mother cells do not die. Only a small segment near the cut end degenerates in a way similar to the distal part. And at the site of injury, the Schwann cells proliferate more actively in the distal stump than in the proximal in an attempt to fill the gap. The gaps up to 3 centimeters may be bridged in this way. Bridging can be facilitated by the stitching the 2 stumps. So here surgery is also required. However, in wider gaps, no grafts may be tried. The distal part of the axons undergoes Wallerian degeneration within a few days of injury. The axis cylinder becomes fragmented and the myelin sheath breaks up into fat droplets. The Schwann cells multiply, which On the other hand, act as macrophages to remove the debris of degenerated axon and myelin, and on the other hand, form a large series of membranous tubes, which play a vital role in degeneration of the nerve fibers. The nucleated cellular cords in the distal terms are called hands of Bergner. So much of discoveries have been made by anatomists as far as this system is concerned. System is of prime importance. Here you can see an image of a normal nerve, image of a compressed nerve, image of a sheath loss with the loss of sheath, and here there is a disconnection also. And here you can see the degeneration. This portion of nerve has degenerated. Okay, and with this, my dear, the JF has gone on, and this is indication for the time to take a break. We'll take a break and we'll come back tomorrow. If not in the morning, in the evening, I'll try to inform you today itself, when next episode will be conducted. And let us take a break today and end our today's webinar here. With this, I thank you for patiently listening to me, and let us take a break today and come back tomorrow. Thank you. Thanks a lot. Have a nice day ahead.